Everyone. In this video, we are going to revise some of the concepts related to the stereochemistry. I'll be uploading such few videos related to the stereochemistry concept so that before we begin with the syllabus related to the MSc Sem 3, we should revise all the concepts whatever you have studied in your previous year. So to begin with, I'm starting with the term chirality and its importance. Now, what do you mean by chirality? Chirality, the exact meaning of this word is handedness. Look at this first image showing you the two hands. So you can look at your own hands. They are mirror images of each other. They are symmetrical with respect to each other. But if I'll try to keep this hand on the top of each other, they won't fit properly. So if you look at this image, if I'll put it on the top of the other, it will not perfectly match unless and until I will rotate this and then I will try to fit it. It will definitely fit. But if I keep this as it is and if I'll try to fit on the top of the first hand, it won't match. So in this concept, we call it as non-superimposable mirror images. Now, if you look at this blackboard, nothing is written on it right now. I'm keeping this in front of the mirror, I will get its mirror image. Now, when there is nothing written, you can see these two mirror images are exactly identical. Now, if I'll keep this blackboard on the top of the previous one, the two images will perfectly fit. That we call it as superimposable. But if you have the blackboard having something written on it, so now look at its mirror image. Now, if I'll keep this mirror image on the top of the previous one, they won't match. So this concept is very important that we have two isomers which are mirror images of each other, but they are non-superimposable. So this concept we call it as the enantiomers. So enantiomers are the mirror images which are having the non-superimposability. Now, this chirality, why it is so important for us, why we are going to study in our syllabus. The reason behind it that in our everyday life, we are going to deal with this chirality. Actually, we are dealing with this chirality. Now, to begin with these examples, let us start with ourselves. Our human body is chiral. It is asymmetric. Now you will say, if I'm looking myself in the mirror, I look symmetric. Then why ma'am is saying that we are asymmetric? The reason behind it, everyone knows that we are made up of proteins. And proteins are made up of amino acids. Amino acids have the two different configurations, D or L, but we are purely made up of L amino acids. Also, everyone knows the sugar which is there in our body is glucose, but we have only the glucose having D configuration. So, if you look at your hands, the way I told you before, they are mirror images of each other. But if you look at their efficiencies, both have the different efficiencies. If you're a right-handed person, you can write with your right hand only. If you try to write it with your left hand, it is very difficult for you. If you are left-handed, you can write it with your left hand only. It is difficult for you to write it with your right hand. That means the two hands, though they look symmetric, though structure-wise they are same, but their efficiencies are different. So like this, there are some compounds in our nature, they identical in the structures, but their efficiencies are different. This is another example. You can see a spinning top. When you see the spinning top, it rotates in a symmetrical fashion. But the path which is following it, it's helical, and helical path is considered as a chiral or asymmetric in nature. Then chirality also important in the case of biology word. This is the best example I can present to you, lemon and oranges. We know that lemon and oranges have the different smell, but the compound which is responsible for this smell is the only one composition, that's chemical composition, is limonene. Oranges contains the R-leminin and lemon contains the S-leminin. But as I said, they have two different smell, but the chemical composition is same. Then mint and the caraway seeds. They have the different flavor, but the compound responsible for this flavor, the chemical composition is same. That is caravone. The mint contains the R-caravone and the caraway seeds contains the S-caravone. But the question is how we can distinguish these lemon and the orange smell. Now we understood that orange and the lemon smell is coming because of the two different enantiomers. But then how your body recognizes it that this is orange smell and this is lemon smell? Because we know that enantiomers, they are chemically identical. Then how come your body recognizes that this is orange smell and this is lemon smell? The main concept that you always have to remember that enantiomers are chemically identical until they are placed in a chiral environment. Now what is that? They are identical if their atmosphere is also identical. But now, for example, as I gave you the example of orange and lemon, let us say you're smelling the orange and lemon together. Your nose is having one receptor, and this nose is having that receptor is enantiomerically pure. Now, what do you mean by enantiomerically pure? This receptor is certain a chemical which is having two isomer, but your nodes contains only one of the isomers. So out of two enantiomers, only one enantiomer is present in your nodes. So we call that as enantiomerically pure. So now when these two compounds come in contact with your nose receptor, which is pure, they are going to have the two different interactions with it. So this R-leminin and S-leminin along with the receptor. So receptor with S-leminin and receptor with the R-leminin. There are two different interactions. So in short, these enantiomers gets converted into diastereomers. Always remember, the enantiomers are identical in chemical properties, but diastereomers have the different properties. This is the basic of your stereochemistry that you'll always remember, because in some of the modules we are going to discuss some of the racemization processes, how to separate the enantiomer. Whichever method you will study, the basic concept is same, that you will first convert your enantiomer into diastereomer, because enantiomers are always chemically identical. When you convert your enantiomer into diastereomer, that time the diastereomer always differ in properties. For example, like they may differ in solubility, they may differ in the adsorption point. So we can use that specific difference and we can separate them. Therefore, always remember the different smell and the taste of the enantiomer is a result of stereogenic interaction in the receptor in the nose and the mouth, and these are chiral in nature. Of course, the chirality is very important in the astronomy world also. Now, when I talk about the sun, it is a rotating sphere and it is symmetrical. But always remember, sun is rotating sphere with a magnetic moment. When the magnetic axis changes its direction, the sun becomes its own enantiomer. You must have heard about this term that called as astrobiology. Now we are doing lots of research on different planets and we are finding the life on the other planet. If you remember, the first life on the earth is we call the that species amoeba, the unicellular cellular species. And when I talk about the cells, cells are made up of the proteins and proteins are made up of the amino acids. So we always say the beginning of the life, it's something the creation of amino acids. So what they are doing research in the astrobiology is that if the other planets have certain condition which can create the amino acids over there, there are chances of life on the other planet. So definitely your astrobiology is somewhere related to your stereochemistry. So all these are very important fields. All we are dealing with in our everyday life, we are coming across with these chiral compounds. But the most important field of the chirality is medicinal chemistry. You go through lots of medicines. You must be using some of the medicine at your home also. Some of uh, them must be using this medicines. Now try to go do some research and see that whether this medicines contain some of the chiral compound or not. And majority compounds you will find they are chiral in nature. They are enantiomerically pure. Taking the right enantiomer is very important when I'm talking about the medicinal chemistry. To prove this point, I'm going to demonstrate you one of the most important example. You can call this as a black day in the field of the medicinal chemistry. This drug thalidomide, this was a wonder drug which was discovered in 1956, and it was marketed in the place, and this was introduced as a sedative, kind of like a mild hypnotic drug. So, later on in few research, it showed that this drug can also be used to avoid the morning sickness for the pregnant woman. And as the results were good, most of the studies were done on the animals. That time the animal testing was allowed. So this drug was tested, but the drawback, or you can say the limitation of that research was your client was the pregnant woman. They were supposed to use this drug on the pregnant woman, but they didn't do the research on the pregnant animals. They did the research only on the normal animals. And once that they got the results, good results with the morning sickness results, they were good. That's why most of the doctors prescribed this drug for the pregnant woman to avoid the morning sickness. But at the end of the decade, that all results came out, and most of the babies who were born in those decades were all deformed. You can see these deformities in them. So they started investigating what was the reason for these deformities, and they found most of the prescriptions were written with the thalidomide drug. Then what went wrong? Like research was done on the morning sickness, all the results were right. Then what went wrong in the research? As I told you, they tested on the normal animals, not on the pregnant animals. Since, see, in the medicinal field, it is very important the kind of the client you're going to select, similar type of research has to be done. So the client was a pregnant woman, but they didn't do the research on the pregnant animal. They did the research on normal animals. That was the first drawback or a limitation. And the second thing, they failed to identify the chirality. This molecule, if you'll see, it's having one chiral center. And when you have a chiral center, definitely there are two isomers which are present. The drug which was given in the racemic form. Now, what do you mean by racemic? Everyone knows that racemic is a 50-50 composition of the two enantiomers. So the drug which was given that time was a 50-50 composition. Out of these two, the R drug, that the R thalidomide, was the good for the sedative, as a sedative or to avoid the morning sickness. But the second isomer was the teratogenic. Now, what is this teratogen? Which stops the growth of the cell. And you can imagine in pregnant woman there is a continuous growth of cell going on, and you have given a drug which is stopping the growth of the cell. Definitely, it causes the deformities. So the main drawback in the research was they failed to identify the chirality. And that's why I'm just emphasizing from the starting that the chirality is very important aspect in all the field, and especially in the medicinal field. If you fail to identify the chirality, it can be a matter of death or life. So that's why it is very important to identify the chirality, to understand the chirality. I will just go a little bit in detail with this incident that later on they did research on this. They separated the drugs. They resolved it. Resolve means resolution means separation of the drugs. So these two isomers were later on were separated, and the proper drug was taken, or proper enantiomer was taken. But further research showed that suppose if you're taking one of the isomer, let's say you're taking S isomer in your body. Once you take the S isomer in body it undergoes the racemization. So it gets converted into its other isomer. So even if you take the right enantiomer, your body conditions makes it allows it to undergo the racemization process. That's why this drug definitely cannot be used as a sedative, because it has a property to undergo the racemization under the human body condition. That's why this kind of research is very important. Even if you say that one isomer can be used, whether that isomer can is it working in your body, finding that is also important in the medicinal chemistry. Right now, if we'll talk about this thalidomide, it is in its right field of the you can say its medicinal property. This drug nowadays is used for the cancer treatment. The cancer, everyone knows, it's uncontrolled growth of the cell, and to stop that growth of the cell, this thalidomide is now used. Just imagine this drug was given to the pregnant woman, and the actual drug or a medicinal property of this drug was to stop the growth of the cell, which was supposed to be used for the cancer treatment. So, if you fail to identify the chirality, a huge mistake in the medicinal chemistry can happen. And that's why identifying the chirality is very important. I hope with this lecture you have understood the importance of chirality, why we should study the chirality, because if I'll find in future you'll be going for more research after MSc, or you'll be working with some pharmaceuticals, or you will be working some chemicals. Over there, you're going to deal with lots of such kind of drugs. And that's why over there, you should have this knowledge of the basic chirality. Thank you. This is a small video, which is I have captured in one aquarium. You can see this is a shell. And if you have noticed this thing, even my first slide was having the shell. Now, what is the connection of this shell with the chirality? I'll just explain you. I'll go to the first slide again. You can see, you have seen many times you blow the shell. This is a blowing side that you just hold near to your mouth. And you can see this is a socket with the help of which you hold that shell. If you'll imagine, let us say, just imagine yourself, you're holding this shell. You can hold this shell with your right hand only, so that this particular blowing part you can take it nearby to your mouth. Suppose you hold this shell with your left hand, this will go away from you. That means if I have to blow this shell, I can hold, there is a only and only way to hold this shell is with my right hand. So this shell is right-handed. It is chiral in nature. So it is one of the best example to present to you. The nature is chiral and we are chiral. Okay, so in future also in some of the videos I'll be covering some basic concept before we start with our lecture. Thank you.